Good morning, good afternoon, and good evening, everyone, and welcome to the 11th event in the 2023 Neurophysiology series, titled Measuring Neuronal Activity and Vascular Physiology in the Human Brain Using High Resolution Functional Magnetic Resonance Imaging, featuring Dr. Jonathan Polimeni, Associate Investigator and Associate Professor of Radiology at the Athenula A. Martino Center for Biomedical Imaging at Massachusetts General Hospital and the Harvard Medical School. Today, he will present his research on the physics and physiology of fMRI and its application to the study of neurological disorders. Just before we begin, we'd certainly like to acknowledge our partners for this series: the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd also like to say a huge thank you to all our series sponsors that have helped to make this fantastic program possible. And now, without further ado, it is my pleasure to welcome Dr. Jonathan Polimeni. Jonathan, thank you very much for joining us, and take it away whenever you're ready.
Great. Super. Well, thanks, Andrew, for this introduction. And thanks again to Dr. Brown and the organizers for inviting me to give this lecture. And so I'm very happy to be part of this webinar series on the topic of neurophysiology and to have this opportunity to talk today about measuring neuronal activity and vascular physiology with functional MRI. And so with this talk, I'll show you how we measure neuronal activity by tracking changes in the blood vessels of the brain, and that these blood vessels reflect neuronal activity remarkably well. And I'll try to convince you that by understanding how brain vascular anatomy and physiology influence hemodynamics at a deeper level, we can extract more meaningful information from our fMRI data. And so in this lecture, I'll address this question: how do brain vascular anatomy and physiology shape or influence hemodynamics and the fMRI signals? Now, all fMRI measurements allow for non-invasive measurement of neural activity across the entire human brain, from cortical gray matter to subcortical gray matter and brainstem nuclei, as well as the cerebellum and even the white matter. However, all fMRI signals that are in use today measure neural activity indirectly by tracking the associated changes in blood flow, volume, and oxygenation that accompany the neural responses, which are collectively known as the hemodynamic response. And because hemodynamics are viewed as being a poor surrogate of neural activity, this aspect of fMRI has often been viewed as the fundamental limitation of the technique applied to human neuroscience and brain imaging. But how much of a limiting factor is this? The blood vessels of the brain are really quite impressive, and the responses are far more precise than many people realize. And so this question is at the center of the research in my lab, and I'll come back to this several times in this webinar. And so first, I was asked to provide a brief review of fMRI for those of you who are less familiar with the method, including some background on the physics and physiology of the fMRI signal. And so I'll begin there, starting with some of the history of the technique. And then after this, I'll shift gears and talk about more advanced topics and ongoing research in my lab. And so how do we use MRI to measure neuronal activity and the associated hemodynamics? Now, the most common fMRI approach measures changes in blood oxygenation. So fortunately for us, oxygen changes the magnetic properties of blood. And so there's, of course, iron in the blood, in the hemoglobin of the red blood cell. And as this audience knows, a hemoglobin is the principal oxygen carrier in the blood. And when it's in its oxygenated state, the oxygen effectively shields the iron in the hemoglobin. And so then when the oxygen is released from the hemoglobin and the red blood cell enters into its deoxygenated state, the iron in hemoglobin is exposed. And so when this happens, the hemoglobin transitions from being diamagnetic like water into being paramagnetic. So incidentally, this property of hemoglobin was discovered by Linus Pauling in the 1930s. So therefore, deoxyhemoglobin is an endogenous contrast agent that can be used to track oxygen delivery in the brain. And the paramagnetic deoxyhemoglobin contributes to increasing the magnetic field in homogeneity where it's present, as depicted in this cartoon, which causes a signal dephasing and a change in the local MRI signal intensity. And so this is the basis of the blood oxygenation level dependent, or BOLD signal, that is used to track brain activity with fMRI. Now, this BOLD technique was first demonstrated in vivo by Seiji Ogawa's team. So what he did was he imaged an experimental animal breathing normal room air, and he could see in the images the venous vasculature where there's a high concentration of this deoxygenated hemoglobin, as seen here, these dark vessels of the brain in this image on the left. And then what he did was to image the same animal breathing 100% oxygen, which saturated the blood with oxygen, effectively replacing all of the deoxygenated hemoglobin with oxygenated hemoglobin, which does not induce any magnetic field offset or MRI signal dephasing. And indeed, the signal in the veins became bright, causing the dark vessels to disappear from the image. And so this is the first demonstration in vivo of the bold effect with MRI. And so the idea is that with the right MRI acquisition that's sensitive to this signal dephasing, we can sensitize MRI to the oxygenation state of blood, and therefore perhaps track changes in oxygenation that occur with neuronal activity and metabolism. Now, initially, after this demonstration in 1990, MRI physicists thought that, sort of intuitively, when local neural activity increased, then metabolic consumption of oxygen should also increase, and we would expect an increase in the local amount of this deoxyhemoglobin, thus a decrease in the bold signal. But in fact, what we see in bold fMRI is exactly the opposite. The bold signal actually increases with neuronal activity. And this is because there's an active hemodynamic response to neuronal activity that delivers fresh oxygenated blood to where it is needed. And so this notion that there is this active response of the brain's vasculature and hemodynamics dates back to the late 1800s, work by Angelo Masso in a surgical patient, and subsequent work by Roy and Sherrington, shown here, demonstrated this empirically. So what Roy and Sherrington did was to drill holes in the skulls of experimental animals and directly measure local blood pressure and brain diameter, and then they electrically stimulated the nerves projecting to those brain regions to generate neuronal activity. And what they saw is that after this electrical stimulation, they saw very large changes in brain diameter and blood pressure accompanying this electrical stimulation. And so this was one of the first direct observations of local hemodynamic changes in the brain responding to neuronal activity. And their conclusion was this kind of famous, often cited quote, the brain possesses an intrinsic mechanism by which its vascular supply can be varied locally in correspondence to the local variations of functional activity. And so this may not be hugely surprising. When the neurons fire, they have more energetic demands and more need for glucose and oxygen for metabolism. So it makes sense that the brain would have the capability to deliver fresh oxygenated blood to the brain regions where it's needed. And so this phenomenon is called neurovascular coupling. And a key question in human fMRI today is, how precise is the blood flow regulation in the brain, and how well do these hemodynamics reflect neuronal activity? So to leap forward about a century later, the first technique that was able to measure brain function across the entire human brain was actually positron emission tomography, or PET imaging, which can label certain molecules like glucose or oxygen with a radioactive tracer, and then measure the distribution of that tracer in the brain following some stimulation or task. And so in this seminal work by Peter Fox and Mark Rakel, they were able to show, in this case, focal increases in blood flow that accompany the increases in neuronal activity, hereby stimulating the motor cortex of the human brain using a simple finger tapping task. And so here we can see that the blood flow increase is quite focal and is localized to the motor cortex, a clear confirmation of the observations of Roy and Sherrington. So now, inspired by these PET results, many MRI groups in the early 90s attempted to use BOLD to map brain function non-invasively in humans. And so here's the first example showing how BOLD MRI can identify active brain regions, performed by Ken Kwong here at MGH. And this photo shows Ken modeling the goggles that he used to deliver visual stimulation to his volunteers in the MRI scanner. So Ken started with MRI data that were sensitive to the bold contrast. You can see these subtraction images that when the stimulus was off, the image was flat, but when the stimulus was on, he saw a big positive signal change in the back of the brain in the occipital lobe, which is the location of the visual cortex. And so he saw that the time course of the signal modulation followed the time course of the visual stimulation, seen here on the right. And so many other groups were able to perform similar demonstrations, including the Ogawa group and Peter Benettini listed here. Unlike PET, the bold contrast is endogenous. It takes advantage of the fact that oxygen modulates the magnetic properties of the blood in a way that can be detected with MRI. And so after these demonstrations, there was a lot of enthusiasm for this technique in the mid 90s, since it provided a new way to measure human brain activity non-invasively. But at the same time, there was some skepticism from the neuroscience community because this measure, based on these regional changes in blood flow and oxygenation, the question was whether tracking blood oxygenation could provide a faithful representation of neuronal activity. And so a landmark study that addressed this key question was led by Nikos Logothetis, published in 2001. And so what Nikos did was to develop a very sophisticated MRI system that enabled him to simultaneously measure neuronal activity or the electrical activity of neurons with microelectrodes and bold fMRI in non-human primates. And so on the left here is a sketch of the electrode that he built that could be used in the MRI magnetic field. And it's a little bit tough to see the bold trace in this plot, so I've drawn in a line on top of the data. What we see is this rapid increase in the measures of neuronal activity, the local field potentials, or LFP, which represent synaptic activity, and the multi-unit activity, or MUA, representing spiking activity, which followed by the bold response. And so what you can see is that the bold response is a little slow. That's because it takes the vessels a little bit of time to open up and the blood oxygenation to change. And it appears to be mostly strongly correlated with the LFP or the synaptic activity. And so this study helped to demonstrate that local neural activity drives local bold responses, and that perhaps the active oxygen supply in the brain, represented by the bold response, mainly reflects synaptic activity. And so with this in mind, now we can consider how neural activity generates the bold fMRI signal. The diagram depicts a vascular tree, the feeding artery at the top, which branches into the capillary bed, and a draining vein at the bottom. And water molecules in the surrounding tissue are the probes in MRI for detecting changes in the magnetic field. And so at baseline, cerebral blood flow supplies fresh oxygenated blood to the capillary bed. Oxygen is extracted at the capillary bed and consumed by the tissue, of course. And as a consequence, venous blood contains this deoxygenated hemoglobin. The deoxyhemoglobin is paramagnetic, as I said, which perturbs the field. Thus, at baseline, there's a rapid signal decay on the venous side of the vasculature, and the baseline MRI signal is low. Now, during stimulation or task, neural activity increases. As expected, there's a somewhat small increase in oxygen consumption, but at the same time, there's a large increase in blood flow. So this is actively mediated by the smooth muscle cells surrounding the arteries that expand, and this in turn causes a passive increase in the blood volume in the downstream veins. So overall, this large increase in blood flow results in an oversupply of oxygenated blood, which kind of flushes out much of this venous deoxyhemoglobin. And this decreased concentration of deoxyhemoglobin on the venous side of the vasculature results in a smaller magnetic field perturbation. And so the MRI signal actually increases compared to baseline. And so this is the basis of the bold fMRI response to neural activity. Therefore, perhaps counterintuitively, the bold signal increases with activation. And as I mentioned a few slides back, one might assume that the bold signal might decrease during activation as neurons consume oxygen and oxyhemoglobin is converted into deoxyhemoglobin, which hypothetically might happen if the vessels themselves did not respond to the neural activity. But instead, in reality, there's this focal and rapid active vascular response that results in this positive bold signal increase with activation. And so the reason for this apparent oversupply of oxygen is still unclear, although several plausible theories have been proposed. It may simply be that for the oxygen delivered by the blood to be able to reach the neurons that are furthest from the vessel, we may just need a high concentration of oxygen in the blood. And so this active vascular response is triggered by a process known as neurovascular coupling, as I mentioned, which is a quite complex and is mediated by multiple signaling pathways. So I won't say much about this today, and these diagrams taken from a recent review article that I recommend highlight some of these complexities. Now, this coupling is still not fully understood and is an active area of research, and recent evidence, mainly from invasive optical microscopy studies, has demonstrated that a perhaps surprisingly tight communication exists between the neural activity and hemodynamics. And I'll show you some examples, some experimental evidence for this claim later in the lecture. Okay, so now there are other ways to manipulate the magnetization of these water protons to make the MRI signal sensitive to other components of the hemodynamic response that can also be used to track neural activity. Cerebral blood flow measurements based on arterial spin labeling can measure changes in tissue perfusion and are thus weighted towards the capillaries, unlike BOLD, which mainly measures changes in the draining veins. It's also possible to measure cerebral blood volume with either with similar techniques like vaso, or by using contrast agents such as gadolinium, which is actually how the first human fMRI experiment was performed, or newer agents such as pheromoxidol, which is what I used to generate the vascular image that I showed in my title slide. However, while these alternate fMRI signals have some advantages over BOLD in terms of their specificity to capillaries and thus neural activity, they have less sensitivity overall, and thus it's more challenging to detect activation in the presence of noise. Therefore, by far, the most fMRI experiments today use the BOLD contrast. Okay, so here's an example movie of some raw BOLD fMRI data playing just a bit faster than real time, acquired while the participant was viewing visual stimuli and performing a motor task. And in these data, the image intensity is related to the local magnetic field perturbations from iron content in the tissue or from deoxyhemoglobin in the blood, with darker voxels indicating more iron content. And the changes we're seeing over time are mainly caused by changes in the local amount of blood deoxyhemoglobin. And so while this movie is showing brain dynamics measured with BOLD, it may be difficult to see any of these activations by eye. And so if I subtract the mean image from these time series data, you can better see the intensity fluctuations in the visual cortex at the bottom of the image and the motor cortex on the left and right sides. But you can see a lot of other fluctuations or noise across the brain that do not appear to be driven by our experimental manipulation. So the goal of fMRI analysis is to separate fluctuations that are driven by our stimulus or task from fluctuations driven by noise, or to pick out the signal of interest out from the noise. And in so doing, identify the brain regions that are responding to our experimental manipulation. And so if you look at the time series data from this one location, we can see that the image intensity is roughly following the on-off pattern of our visual stimulation, just like we saw in Ken Kwong's original study. So in fMRI analysis, we compare the sort of idealized pattern of neural activity based on our known stimulus or task timing with the recorded intensity fluctuations to infer which brain regions are responding. So in fMRI experimental design, we must make sure that we compare two conditions that allow us to identify which parts of the brain are responding to the feature of interest. So this is a classic example from Nancy Kenwisher's lab in which she showed participants pictures of faces and pictures of objects and found a small region of the brain where the voxels are responding more to the pictures of faces. And so to confirm that these voxels were really selected for faces, she repeated the experiment, but in this version compared the faces to cut up and scrambled versions of the images, which had similar spatial frequency content, and again found that this region only responded to the intact face images. Now, this experiment has been replicated many times, and this finding of a face area in the fusiform gyrus is quite robust and provided more evidence for a modular theory of brain organization under which different brain regions have distinct roles in specific sensory, motor, or cognitive tasks. And this is a famous example of how fMRI has contributed to human neuroscience. And so while fMRI today is heavily used for human neuroscience, it's also used clinically and is trusted enough to be reimbursable by health insurance companies. And so this example insurance company lists in its policy that fMRI is medically necessary to identify the eloquent cortex in pre-surgical evaluation of persons with brain tumors, epilepsy, or vascular malformations, and is therefore covered and reimbursable. And several clinical research groups are working towards expanding the use of fMRI for diagnosis and treatment monitoring for both neurological and psychiatric disorders. So finally, fMRI can be used to infer brain connectivity. The idea is that even in the resting state, there's always spontaneous neural activity that creates random looking fluctuations in the BOLD signal. And if these fluctuations are measured in two brain regions are correlated, which is unlikely to occur by chance, one can infer that it's likely that some anatomical pathway connects these two brain regions, and they are said to be functionally connected. And so if we extract the time series data from a seed region and then correlate that with the time series data across the rest of the brain, we can estimate a coherent network of brain regions as shown here. And And there are many such standard brain networks that are robustly observable across individuals, such as the network of brain regions that is more active when the subject is not engaged in any particular task, known as the default mode network, as well as many other networks shown here. And so there can be fMRI signal fluctuations driven by non-neuronal sources, which I alluded to previously, such as the respiration or cardiac cycles, or even subtle head movements. And especially in resting state data, where the spontaneous neural fluctuations are subtle, one must be careful to address these so-called physiological noise sources when estimating functional connectivity. And so today, estimating functional connectivity and brain networks is one of the main applications of human fMRI. Okay, so with this background, I'll switch gears and talk more about some advanced topics and ongoing research in the lab. So this will be an outline of the topics I'll briefly cover on this sort of general theme of how vascular anatomy and physiology influence fMRI, and I'll keep this fairly high level. So first, I'll talk about high spatial resolution fMRI and describe what we call anatomically informed analyses of these high resolution fMRI data. And there I'll pose the question, can these analyses help to improve the sensitivity or specificity of the bulb response? Then I'll talk about high temporal resolution and show how fast fMRI techniques can track surprisingly high frequency neuronally driven oscillations in the bulb response. And there I'll ask, how sluggish really is the hemodynamic response to neural activity? Then I'll show some ongoing work developing biophysical models of fMRI responses and ask, can realistic vascular models provide us insight into the observed bulb signal? And then I'll conclude by showing an example of physiological MRI of a vascular reactivity and ask whether standard fMRI can be used to measure impaired neurovascular physiology. Okay, so first, high resolution fMRI. And so as I mentioned earlier, a fundamental question in fMRI is, how well do hemodynamics reflect neural activity? And a related question is, what's the ultimate or achievable resolution of fMRI? So this is relevant for those of us who wish to map fine scale units of functional architecture, such as cortical columns, layers, and small subnuclei. And so to address this question, we first should distinguish between two concepts, imaging resolution and biological resolution, which I'll define. We've seen that advances in MRI technology provide us with smaller voxels and faster sampling rates, thus increase our imaging resolution. So here's an example from Avery Berman from our lab showing a record-breaking high imaging resolution, 0.6 millimeters isotropic fMRI, covering the entire brain at 7 Tesla, which required a lot of effort to achieve. And so as a point of comparison, most fMRI studies today use 2 to 3 millimeter voxels, and so Avery's voxels are orders of magnitude smaller in terms of their volume. Now, with this capability, a natural question arises. Is the accuracy of fMRI today really limited by voxel size or by what I like to call the biological resolution of the technique, by which I mean the precision in space and in time of blood flow regulation in response to neural activity? And so conventionally, biological resolution is thought to be coarse in space, maybe 1 to 2 millimeters, and sluggish in time, with hemodynamics peaking maybe 6 seconds post-stimulus. However, our colleagues, mainly in the field of optical microscopy, have shown us that fundamentally, the biological resolution of any hemodynamics-based measure is far better than previously believed. Now, this biological resolution is thought to be limited by the spacing and timing of the control structures of blood flow, which were presumed to be the smooth muscle cells that surround arterioles, and maybe these are about a millimeter apart or so in the human brain. However, mounting evidence suggests that pericyte cells that have been a long time to wrap around capillaries are capable of controlling capillary diameter. So here we can see a movie of some pericytes in action. You can see a single capillary with individual red blood cells here. In the middle is an electrode attached to a pericyte, and when the electrode stimulates that pericyte, it constricts, showing that it's contractile and controlled diameter, which I think is super cool. But perhaps what's more amazing is that, and this movie loops in case you missed it, that just after the stimulated pericyte to the middle constricts, the neighboring upstream and downstream pericytes, indicated by these red arrows, also constrict. So this shows us that there's some coordination among the pericytes, and we know that there is signaling or communication along the endothelial cells that line the vessel walls. So this tells us that there is machinery in place for varifocal blood flow regulation. The bad news may be that there's some coupling of the diameters between multiple capillary locations, which may constrain or limit the degrees of freedom of blood flow regulation. Now, these remarkable data were acquired in vitro in excised retina, where blood pressure and flow weren't at physiological levels. However, the same group revisited this years later and showed that pericytes are able to actively control capillary diameter in vivo as well, and that this capillary dilation actually precedes the upstream arteriolar dilation. So the dilation seen at the capillaries is not a passive response to any upstream flow or pressure changes. Now, while there's still a debate over whether the parasite cells observed in this study were really on capillaries or perhaps on precapillary arterioles, and some disagreement whether all parasites are contractile, either way, these data show us that there are mechanisms in place for very tight blood flow regulation at fine spatial scales. And so, briefly, while these data were all taken from small animal models, there is recent evidence that parasites play a similar role in human brain as well, and therefore they're relevant for our understanding of the biological spatial resolution of human fMRI. Okay, so now considering temporal resolution, in this study conducted by Anna Devore's lab, they measured in mouse somatosensory cortex calcium dynamics in astrocytes concurrently with neighboring single blood vessel dilations in response to four-paw stimulation, and found that while astrocytes activate about 3 seconds post-stimulus, the vascular activation onset occurred on average 0.7 seconds post-stimulus, and sometimes even faster. And so not only do these data question the role that astrocytes may play in neurovascular coupling, but they also teach us that the smallest vessels, if we can see them, respond very rapidly to local neuronal activity, again suggesting potential for perhaps surprisingly tight blood flow regulation and a high biological temporal resolution. And so if we consider these results together, they suggest that the biological resolution is likely to be higher than the imaging resolution we can achieve today, even with modern fMRI. And so this motivates us to develop technologies that can enable smaller voxels and even faster acquisitions. Although today it's still unclear what the ultimate biological resolution is, and it's likely to vary across brain areas. It's perhaps, it appears to me tighter than 0.6 millimeters. And so voxels smaller than what we have achieved would be put to good use. And so the question then becomes, how do we leverage this intrinsically high biological resolution? And so one strategy begins by recognizing that there's this hierarchy of vascular anatomy in the cerebral cortex, which can be appreciated from these beautiful electron microscopy data from monkey primary visual cortex. So on the surface of the cortex, we have these large PO veins that drain deoxygenated blood from large cortical domains, then the ascending venules running perpendicularly across cortical layers, and here the parenchyma, the seemingly randomly oriented capillary bed. And the idea is that as our fMRI voxels become smaller, much smaller than the thickness of the cortical ribbon, we can exploit this vascular organization and, for example, sample exclusively from voxels at the capillary bed where the coupling between the hemodynamics and the neural activity is the tightest, while at the same time discarding voxels, sampling the large veins that carry these non-specific fold responses. And so this is an example of what I call an anatomically informed analysis. And so to test this, we utilized the well-known retinotopic mapping in the human primary visual cortex or cortical area V1 to generate a visual stimulus that would elicit a ground truth neuronal activation pattern in the shape of the letter M in V1, as shown here. And then we sampled our small fMRI voxels across the cortical depths to test how the activation pattern changed when sampling either close to or away from those large PO veins. And so here is the activation pattern on the reconstruction of the cerebral cortex, shown nicely in the so-called inflated version of the surface. You can see that it's tough to see the M activation tucked inside the calcarine sulcus in the folded representation, but if we inflate the cortical surface reconstruction, we can better visualize this activation pattern. Okay, so we can use knowledge of what the spatial pattern of neural activity is supposed to look like to evaluate the bold fMRI data. And so if we analyze the fMRI voxels that are closest to the white matter surface and project these onto this inflated cortical surface reconstruction, we can see that the spatial fidelity of this pattern is actually pretty good. But as we progressively sample from voxels that are sitting closer and closer to the peel surface, the spatial fidelity sort of breaks down systematically, but I think in an informative way. And so, for example, we see this artifact at the peel surface that may be a vein that's just cutting off the top corner of the M. So this tells us that the biological resolution of fMRI is complicated and does not appear to be a simple Gaussian point spread, and the pattern observed at the peel surface is not simply kind of a smoothed version of the pattern that we see at the white matter surface. This is because our fMRI voxels are really small and are really sensitive to the local details of the vascular anatomy. Now these are old data, and I'm including this here today because this was kind of a fun experiment that showed us in a concrete way something that many had suspected about those large draining veins at the peel surface. However, when I first presented these results about, 14, 15 years ago now, the community had a hard time accepting that the biological resolution of fMRI was even high enough to even see this pattern based on the understanding of the time, and the field has come a long way since then. Okay, so we can use this analysis approach to look at the bold activation strength across cortical depths. Cortical depth is basically a proxy. At deeper cortical depths, the voxels are far from the big draining veins, but at shallower depths, the voxels will contain those big draining veins. And so here's the activation strength at the deeper cortical depths near the white surface, and here it is at shallower cortical depths near the peel surface. We see that the percent signal change of the bold response is about twice as high near those large veins than it is at the white matter, which makes sense because, again, there's more deoxyhemoglobin in those veins, and so we see a bigger bold signal change there with the activation. And so if we consider this alongside the activation maps, together these plots tell us that, kind of sadly, the sensitivity is highest exactly where the specificity is lowest. That's at the peel surface. And so this trade-off means that for columnar systems like retinotopy, where the neural activity is really the same across cortical depths, we have a knob that we can turn during the analysis. If we need more sensitivity, we can sample near to the peel surface. But if we're prioritizing neuronal specificity and spatial accuracy, we should sample near to the white matter surface. Okay, so it appears possible that human fMRI can achieve high resolution. And we expect to be able to achieve this specificity in part because classical studies have shown us that fine scale features of functional architecture, known as cortical columns, can be resolved with hemodynamics based intrinsic signal optical recording. And so this method exploits the fact that, so not only do the magnetic properties of blood change with oxygenation, which is the basis of the bold signal, the optical properties do as well. And so the reflectance of different wavelengths of light from the exposed cortex seen through a cranial window can be used to measure oxy and deoxyhemoglobin changes. So here one can clearly see here in the exposed macaque monkey visual cortex, columnar features well below 1 millimeter in size. For example, the iso-orientation domains in the middle here, and the classic ocular dominance column or ODC zebra stripe pattern shown here on the right. And so while there are some differences between this signal and the bold signal, these maps are still based on hemodynamics and concretely demonstrate that the biological resolution of these hemodynamics is actually quite high. And so if we can achieve small enough voxels, we expect to see columnar systems using human fMRI as well. And indeed we can. So evidence for ocular dominance columns in humans has been provided by human fMRI for about 25 years. But perhaps the best known example is from Isa Yacoub, who showed that spinnacle bold, which is kind of a tricky to use variant of bold that's less sensitive to these large veins compared to standard gradient echo bold, can be used to resolve this ODC zebra stripe pattern in humans. And so we later demonstrated that it's possible to image this ODC pattern with standard gradient echo bold over a large region of V1 using our anatomically informed sampling approach. Here, our isotropic high resolution acquisition and our surface-based analysis allowed us to avoid those peel veins and to see this pattern over large regions of folded cortex. We're no longer restricted to locally flat patches of cortex as in those previous studies. And so this anatomically informed sampling provides us with a huge advantage. Since gradient echo bold has higher sensitivity, than the tricky spinnacle bold variant, even for voxels that only contain capillaries. And this is important because the signal changes used to map out these ODCs, they're really tiny, and here we need all of the sensitivity that we can get. Okay, so we've used this approach to look at columnar systems outside of primary visual cortex or area V1, such as the area V2, thick, thin, pale stripe system. And in this case, I'm showing only the color selective thin stripes. So you can see these columnar stripes extending sort of perpendicularly from the V1, V2 border here in the inflated representation on the left and on the flattened representation on the right. So now if we sample this activation in the superficial gray matter near to those peel veins, we cannot resolve these stripes, but if we sample deeper, closer to the white matter, the columnar pattern becomes more distinct. And so this again shows how this anatomically informed sampling can provide the improved spatial resolution and allow us to see these fine scale patterns of activity. Okay, so next I'll talk about fast fMRI and biological temporal resolution. And so this is work that was led by Laura Lewis, who wanted to see whether we could track fast, bold responses to oscillating visual stimuli. So we're mainly interested in endogenous oscillations, but in this case, we induced neuronal oscillations in a known waveform and frequency using visual stimulation, consisting of a standard Gaussian checkerboard whose luminance contrast was modulated at several temporal frequencies of interest. And to track these bold responses, we acquired fast fMRI data with a short temporal sampling interval of about 250 milliseconds. And so if you consider the predicted bold responses to the stimulus using what's known as the canonical hemodynamic response function, which is assumed by most fMRI studies to capture the temporal behavior of the bold response, we see that at low stimulation frequencies, the predicted bold response clearly tracks the stimulus. However, as we move to higher and higher oscillation frequencies, to the point that the oscillation period is short compared to the temporal width of the hemodynamic response function, we see that after an initial transient, the bold response oscillates around a new baseline or plateau, and the amplitude of the oscillations around this plateau kind of gets smaller and smaller with higher frequencies, kind of exponentially, but never quite reaches zero. And so the question is, what oscillation frequencies are we able to detect with our fMRI data? And so in these trial-triggered averages, you can see that the bold signal is able to track both 0.2 hertz oscillations as well as oscillations as high as 0.75 hertz at 7 Tesla. So this was a far higher frequency than we had imagined that we could detect. Now note the vertical axes in these plots. The bold response to the 0.75 hertz stimulus was really tiny, but detectable, yet it was still 10 times larger than what we predicted using this canonical hemodynamic response function. This means that the canonical hemodynamic model just does not generalize to such rapid stimuli. And bold fMRI can detect these rapid oscillations, but their amplitudes are just very tiny. And we know that the hemodynamic response exhibits some nonlinearity when using short duration stimuli, which I don't have time to explain today. However, this nonlinearity could potentially explain our observations. The bottom line is that with the right experimental design, fMRI seems capable of tracking surprisingly fast neuronal oscillations. And finally, similar stimulus-driven bold oscillations have been reported in motor cortex and somatosensory cortex in this collaborative work with Shota Hadono and Martijn Kloos at the University of Queensland. Here, using a data glove that records hand motion during MRI scanning, they also measured bold responses tracking up to 0.5 hertz activation, with again, amplitudes far higher than what were predicted from the canonical model. And they also examined the response cortical depths and found that at low frequencies, the bold response is largest near the peel surface. But at higher frequencies, including 0.5 hertz, the depth profile kind of flattens out, suggesting that these large draining veins at the peel surface just can't keep up with these fast oscillations. And the small vessels within the parenchyma are relatively fast, suggesting that the capillaries may not only exhibit improved spatial specificity, as I showed you a few slides back, but perhaps improved temporal specificity as well. Okay, so to summarize this portion, we have shown that bold fMRI can track oscillatory neural activity up to about 0.75 hertz. We have some preliminary data suggesting that we can perhaps see up to 1 hertz. So this suggests that the fMRI signal can reflect meaningful neuronal dynamics approaching the timescales of many cognitive processes. These data also imply that This canonical hemodynamic response function, originally derived from long stimulus blocks, may not be appropriate for capturing the bold response to shorter stimuli or to subtle or ongoing cognitive processes or more naturalistic stimuli. And we're currently beginning to apply biophysical modeling to better understand these fast hemodynamics and nonlinearities. And so next I'll describe our first principle biophysical models of fMRI. And so the biophysical models that I'll be discussing are based on both realistic microvascular anatomy and dynamics. And this bold modeling approach was pioneered by David Boas and his group. And so we call this approach the vascular anatomical network, or VAN modeling approach, as it differs from other classic bold modeling approaches, such as the balloon model that some of you may have heard of. So in a bit more detail, this approach was developed by our colleagues in optical imaging who used two-photon microscopy to image the full vascular network, essentially all vessels within an fMRI voxel at one location in the cortex of a mouse, consisting of a handful of input arteries and output veins and a couple thousand capillary segments. And so with this, we can simulate the full spatiotemporal dynamics of the hemodynamic response. The input is a time trace of feeding artery diameter changes, which reflect the vascular response to neural activity. And then we use computational fluid dynamics to simulate local blood pressure, blood flow, volume changes, blood oxygen advection and oxygen diffusion into the tissue. And then we can compute the magnetic field offsets in and around the vessels, and finally the MRI signal, which gives the bold response. And so these models differ from previous approaches because here the vascular anatomy is connected as a single network, which allows us to simulate the dynamics of the microvascular response, as shown in this animation, and understand how both the geometry and topology of the microvasculature kind of shape the bold fMRI response. And so while this approach was developed for mouse cortex, our goal has been to build a VAN model for the human cortex. The main challenge with VAN models are the inputs. We require accurate microvascular network and accurate microvascular dynamics as inputs. And while it's possible to reconstruct the full microvascular network in vivo in small animals using invasive microscopy, it's of course more challenging to reconstruct the full 3D vascular network in humans. And so for this reason, to generate a human VAN model, we instead synthesize the VAN based on vascular distributions taken from the literature. So here you can see an example, a synthesized network of one fMRI voxel in one location of the human cortex next to a reconstructed network of one location of the mouse cortex to give you a sense for the scale. Now, to validate our synthesis approach, we first generated several synthetic VAN models that are matched to existing reconstructed VANs in mice and computed bold responses from both. And the results show that synthetic and real VANs yield similar bold responses, as shown in these responses at the bottom right here, with similar timing and amplitudes, which is a good sign that suggests that the synthetic VANs behave similarly to the real VANs. Okay, so coming back to our proposed human VAN model, we know that mice and humans have different vascular architecture, both in terms of, both in terms of the topology. For example, the artery to vein ratio in mice is nearly the opposite to that of humans, and the geometry, both diameters and lengths of the vessels are very different between mice and humans. And so we wanted to see whether mouse versus human vascular architecture impacts the bold timing. Indeed, we found that partly because of the longer vessels in humans and the corresponding longer time it takes for the upstream propagation of arterial dilation, the bold response from the human VAN, shown in this blue curve here, was slower than that of the mouse VAN, which matches with measurements of the bold response in these two species, showing how vascular architecture may influence the hemodynamics. And so I won't go into the details today, and this needs further validation, but it shows us how VAN modeling can generate potential insights and new hypotheses that we can test empirically. Also, most of what we know about neurovascular coupling and microvascular hemodynamics comes from mouse data. And so it will be important for us to identify ways in which hemodynamics may be different in humans in order to properly interpret our fMRI data. Okay, so I'll wrap up by sharing an example of our work towards a physiological MRI of vascular reactivity. And as many of you know, there's increasing evidence suggesting that vascular pathology plays a major role in many brain disorders associated with cognitive impairment. And this white paper, published this summer in Stroke by Idiacola et al., makes the case that several hemodynamic measures are associated with cognitive function and calls for the development of more non-invasive biomarkers of neurovascular dysfunction. And so the tools that we are developing for fMRI to track neural activity can therefore be repurposed and be helpful for understanding vascular disease. And my first involvement in this effort was over a decade ago when we applied bold fMRI to investigate reduced vascular reactivity in cerebral amyloid angiopathy. So cerebral amyloid angiopathy, or CAA, is a disease that co-occurs with Alzheimer's disease, in which the same amyloid plaques known in AD accumulate in the parenchymal blood vessel walls and hinder smooth muscle cells, causing impaired reactivity and some neurovascular uncoupling. And so in this first study, led by Steve Greenberg here at MGH, who's a renowned expert in CAA, we demonstrated that the time to peak, time to baseline, and amplitude of the bold response to flashing checkerboard visual stimulation all differed in CAA patients compared to the age-matched controls. Overall, the bold response was slower and weaker in CAA. Now, the baseline perfusion, however, was the same across groups. We saw no signs of weaker neuronal activity, and so we interpret this as abnormal bold dynamics reflecting vascular pathology. So since then, our results were replicated by several groups, and because this measure was so robust, bold response dynamics were used as an outcome measure in a drug trial for a CAA treatment developed by our neighbors at Pfizer. And so we provided several sites involved in the trial with our flashing checkerboard stimulus and some basic fMRI analysis tools. And indeed, we saw that the treatment did result in a measurable change in the hemodynamic response. And so this shows us how standard fMRI experiments, rather than being used to measure neuronal function, can be used to investigate abnormal vascular physiology. And because the vascular dynamics appear to play a role in the glymphatic system via neurovascular coupling, we're beginning to apply these physiological MRI measures to understand brain clearance as well. Okay, so finally, just to wrap up, I wanted to show some preliminary results of some ongoing work quantifying stimulus-evoked blood velocity responses in major supplying arteries measured with so-called functional magnetic resonance angiography. So this is work by Jonathan Hu in my lab, who's using an MRI angiography technique that's usually used to measure baseline blood velocity. He adapted it to measure changes in blood velocity accompanying neural activity to provide a more quantitative measurement. So here he's looking at velocity changes in a branch of the posterior cerebral artery, or PCA, which supplies the visual cortex. So here you can see the activation displayed as a heat map overlaid on the raw image using a standard analysis similar to what we use for bold fMRI applied to these velocity data. So the activations are sparse, of course. We detected strong responses in the right and left PCA, shown here, and maybe some hints of a response even in the middle cerebral artery, whose branches supply some portions of the visually responsive cortex in the occipital and parietal lobes. And so here are the response time courses. With an inset zoom in of the activation map, the velocities increase from about 28 to 32 centimeters per second, or by about 20%, which was a surprisingly large increase to me, but it is comparable to what has been reported previously with invasive Doppler measurements in humans. And so this is a substantial velocity response in vessels that are about 6 centimeters away from the visual cortex, which I find remarkable. And so we're now trying to see how far upstream we can detect these blood velocity changes. Now, it's not yet clear how far upstream we would expect to see these velocity changes. And while what we're seeing could be a mix of neurovascular coupling and autoregulation, and this audience may know a lot more about this than I do, it shows us how MRI can be used to address this question in humans, and how we're discovering new ways to extract quantitative physiological information from the neurovasculature that can be used to inform our biophysical models and also provide more insights to the mechanisms of brain hemodynamics and fMRI. Okay, so to recap, I provided some background on bold fMRI and reviewed how it reflects an active vascular response to neural activity. I've described how anatomically informed sampling A bold fMRI data can, in some cases, improve neuronal specificity. I've shown you how the bold response tracks high temporal frequency activation and exhibits high temporal specificity, and that we're able to detect much faster responses than we had imagined. I've also shown how, at small spatial scales, biophysical models can provide insights into microvascular influences on the fMRI signal and how can perhaps help us to understand the ultimate biological resolution of fMRI. And then I closed by showing an example of how fMRI tools that were originally developed for measuring neuronal activity can be applied to measure vascular physiology. Okay, so just to conclude, we're still learning about how the vasculature shapes or influences the fMRI signals. And although the ultimate biological resolution of fMRI is still not known, there is, of course, a limit, but there's no evidence to suggest that we've reached that limit yet. And so our vision is to combine a high imaging resolution with models of the vascular architecture to develop physiologically informed analyses and interpretation of our fMRI data. And so I think that overall, there's a lot of untapped potential in fMRI. And I think that a deeper understanding can provide us with enhanced specificity and new insights into human brain function as well as vascular health. And so with that, I'd like to thank the following people, especially Laura, Grant, Zhongshen, Shaheen, Doug, and Shota, as well as Professors Anna Rowe and Bruce Pike for sharing slides. My lab, our funding sources for their generous support. And of course, thank you for your attention.
Thank you, Jonathan, for that wonderful presentation. That was really excellent. Thank you. Ready to jump into some questions, John?
Sure. Yeah, anytime, Andrew.
Okay, great. One here, I think, is a great one. If parasites are able to regulate blood flow of capillaries, then
Mhm.
why is their ultimate the ultimate biological resolution not understood? And what is the actual spacing between parasites in the human brain?
Yeah, so no, yeah, this is a question that I do see a lot. And so, yeah, parasites seem capable of regulating blood flow, but recent evidence suggests that they may act somewhat slowly in response to neural activity, and so the jury is still out on whether they're really participating in neurovascular coupling, or maybe they're mainly there for more kind of slower changes, homeostatic changes, adapting blood flow kind of on a slower timescales. And so it's still the role of parasites in neurovascular coupling is still unclear. I think what's I think important to note is that they do represent a mechanism. They do represent some way in which blood flow could be regulated at very fine spatial scale. So again, their role in neurovascular coupling is still not entirely worked out. Also, as I mentioned, this parasite activity seems to be sort of coordinated. As I mentioned, they seem to coordinate with one another. So the degrees of freedom in blood flow regulation at these kind of small spatial scales may be limited. Now, and so yeah, it may not be completely dictated by their spacing. So I'm not sure what the spacing is in humans, but from the images I've seen, they're somewhat sparse, maybe spaced about 50 or so microns apart. But that might be in mice. I can look into this and post a few papers on this topic that would have a more definitive answer. But yeah, we're still trying to understand the exact role of these parasites, but they do tell us that fundamentally there are mechanisms in place for very focal blood flow regulation.
Okay, fantastic. I'm not sure if I fully understand this question. Perhaps you will. Has there been any evidence of functional cerebral sympatholysis in health and disease states?
Oh gosh, yeah, maybe I don't understand the yes. So maybe I don't understand functional. Let's see, I'm not familiar with this, and so I'm apologizing. I'm not able to. Oh, sympatholysis. Yeah, apologies. I guess I'm not qualified to answer this, but
Not a problem. Uh, I mean, you talked a lot. There's a great question here. A common one, I think, with MR,
mhm.
with fMRI. How long does it take for the various acquisitions? And
Oh.
for clinicians, how much time should you ask the patients to devote to get the required data?
Oh gosh, that's a great question. I mean, so what I would say is, obviously, the more the better. It depends on the patient population. I think that there are new tools for online QA/QC of the data, so that if you have a patient population, you're running a 6-minute scan, and there's some movement during the scan, the techs can be, alerted immediately. You can give the patient a pep talk and repeat the scan. So I think that these online QA/QC tools are really valuable so that not only can one repeat if needed, but one knows when enough data have been acquired. I think that it really depends, however, on the paradigm. I think that, for example, one can see the default network in resting state fMRI in about 10 minutes of scanning, and sometimes even less. Visual activation, for example, in this multi-site trial that we did for the CAA drug treatment, I think there was about there's maybe two runs of about 6 minutes each. That was also very robust. I think the important thing to remember though is that while longer scan times mean more sensitivity through averaging, um, it's often important to recognize that it's not just the sensitivity to the main response, but it's the sensitivity to detect differences between the groups, and sometimes that contrast, how much of a difference in the bold response between groups are we expecting to see? That sometimes is hard to know ahead of time. And so what I'd say is if the scans are too long, there will be behavioral changes. There can be fatigue, cognitive changes, and so we try to make the scans as short as possible. It really depends. I think that some patients can handle 30 minutes. Some patients are more robust. Yeah, it's hard to answer that question definitively, but these are some of the questions I would ask when trying to determine this for any given population.
Right. Yeah, that's fantastic. Uh, a question here. What is your opinion about cerebral vessel changes in people or animals with metabolic syndrome? You know, factors such as vessel bubbles, microaneurysms, vascular anastomoses, etc.
Oh, that's interesting. We've actually just started to look into this, and I guess as you saw from my final slides, I was a bit surprised, to see these vascular changes, so far upstream from the site of neural activity, um, in our kind of in the PCA, we are seeing much larger responses than I expected. So I guess what I would say is my expectations may not be, so um, informative. I mean, I guess what I would say is we do know that, for example, with these anastomoses, I mean, we do see that these do impact the blood flow. We're starting to understand better how the vascular network architecture, um, how there are emergent vulnerabilities and resiliencies in blood flow patterns that are based on the network topology. And so I think that based on these kind of preliminary observations, I would expect, the vascular topology and anatomy to shape these hemodynamics. But I think that I'd have to see, Andrew, what were the other examples?
Uh, the other examples were anastomoses, vascular anastomoses,
Osmosis.
vessel bubbles, microaneurysms.
And microaneurysms as well. We've looked into this a little bit. They also do divert blood flow patterns, but it's tough. I don't have data on this. I would guess that, again, there would be some influences, but it would really depend on where in the vascular network the aneurysms are located.
We are at the hour, but if you have a moment to continue, John, if you have the time, to get through the last couple questions, is that okay?
Oh, I'd be happy to, and
Okay.
also I'm happy to answer other questions offline. Especially these are some good ones. I might have to think a little bit, and maybe I can respond more fully offline.
Wonderful. Yes, and if you do have any questions that occur to you afterwards, please add them in the survey. We'll forward them on to Jonathan as well.
Great.
Along the lines of that last question,
Mhm,
are there any there's always surprising to me the
mhm.
extent of heterogeneity between different brain structures, vascular
Yes.
architecture between individuals, and so on. Are there
Yes.
conditions that
Yes.
preclude application of fMRI? Can you learn more information perhaps from these unusual outliers? Are there disease conditions that preclude application of fMRI?
Oh gosh, I love it. I mean, I guess what I would say is yes. I mean, well, there's a couple of questions there. We can learn a lot from looking at these outlier cases, and we have learned a lot. It's sort of remarkable to me how we have more work in these CA patients. We've actually learned a lot about the regulation of blood flow in healthy subjects by seeing how it's affected in these disease groups. We've actually learned a lot about, again, like, the propagation of dilation, for example, from the parenchyma to the feeding arteries by seeing how it's altered in disease. So I think the answer is yes. Now, in terms of precluding, I think that, you know, I referred to the CEA, this reduced vascular activity, as a form of neurovascular uncoupling, and maybe that's a bit of an extreme viewpoint. I mean, we do see that there's a delay in the hemodynamics, whereas there's still a coupling. It's just a kind of an impaired coupling. In other cases, famously in tumor patients, we do see a much more severe uncoupling between the hemodynamics and the neural activity. And so, what's important, I wouldn't say that that precludes the use of fMRI. As I mentioned, fMRI is reimbursable for pre-surgical mapping in tumor patients. What we have to be mindful of is that if the relationship between the neural activity and the vasculature is different or impaired, that this could influence our observations. It might result in false negatives. And so there are techniques such as cerebrovascular reactivity mapping that we use to kind of map out locations in the brain where there may be some uncoupling. And so CPR, or cerebrovascular reactivity, can be performed with the CO2 administration, or even clinically with a breath hold, because CO2 is a potent vasodilator. We do use this to see how the vessels across the brain are reacting. And if we identify locations in which there is reduced reactivity to CO2, we know that in those locations, these bold signals will be, have to be interpreted with care. And so what I'd say is that I don't think we've come across any cases where the disease would preclude the use of fMRI, but we do have to be mindful of, how the disease affects the hemodynamics, and to build that into our interpretation. And like I said, these techniques like CPR are becoming much more routine in order to do so.
Right. Wonderful. Um, Question. I think this is one you could potentially answer relatively quickly. Fantastic presentation. For how long does the coordination of parasites persist?
Ah,
Um, I think your video showed 3 parasites, but does it extend even further?
oh, yes. Yes. So there's a really nice paper by Andy Shee's group, Hartman et al. I think it was 2021. They showed so that movie I showed, I think, is gorgeous. It was from 2006. It really showed very concretely the action of parasites. But since then, there's been a lot more work on this, and I think this really nice work by Andy Shee shows that the coordination extends much further. Um, I hesitate to ascribe a concrete number because I don't quite remember his results, but yes, and as I said, the good news is that the mechanisms for this coordination are also pretty well understood. Also, recent work from Mark Nelson's group has been showing the communication amongst these parasites, and so yes, it appears to extend much further than what was shown in this beautiful movie from David Atwell's lab that I showed in the presentation.
Wonderful. I think, just in the interest of time, to get everyone onto their next appointment, maybe we'll make this the last one.
Mhm.
How do you compare and contrast the clinical applications of
Yeah.
fMRI versus optical fiber-based approaches, such as long wavelength interferometric diffuse correlation spectroscopy? Differences are vast, but just curious to hear your opinion.
Yeah, differences are vast. I guess what I would say is that, I mean, what I like about fMRI is that, again, unlike other neuroimaging techniques, it really allows us to look across the entire brain. I mean, we use fMRI for looking at responses in the deep cortical areas, brainstem, thalamus, C-spine even. And so I think that fMRI does provide, fairly uniform sensitivity and specificity across the entire brain. Now, I think that, we do see a lot of value in multimodal imaging. I think that, we've used, for example, concurrent optical imaging, you know, um, near infrared spectroscopy, for example, with fMRI to get a better picture. I think that the two can be complementary. These optical techniques have a much higher temporal resolution and more specificity to different components of the hemodynamic response. But generally, I think that of all the tools we have available to us today, fMRI at least provides the best coverage across the brain and sensitivity across brain structures at pretty reasonable spatial resolution. And as you're seeing, we are certainly improving the temporal resolution as well. It's going to be a while before we reach the temporal resolution of optical, maybe never. But I do think that these techniques are complementary, and depending on the brain region of interest, one may be more suitable than another.
And my understanding, my impression has been that the trend towards, an increasing number of MR compatible pieces of
Yes,
equipment is certainly growing as well, right?
yes, 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 absolutely.
Uh, I think, unfortunately, John, I think that's all the time we have for today. But I really wanted to thank you for being with us and sharing all your insights. It's been fantastic having you here.
Great, it was a lot of fun. Thank you, Andrew. Thank you,
Thank you very
everybody.
much. Another big thank you to our partners for helping make this series possible. And in closing, we hope you enjoyed this Insight Scientific webinar, and we'll see you again next time. Thank you, everyone.